Hi everyone, this is Ms. Romani, and for this lesson we will be learning about enzymes. And by now you may be a little familiar with enzymes. During our lesson on proteins, I briefly mentioned that enzymes are responsible for the chemical reactions in living cells. So just for fun, I tried looking up how many chemical reactions are actually happening in a human body every second. And not surprisingly, there is really no definitive answer to this question. First, I found out that the number of chemical reactions happening in a single cell is huge and varies tremendously depending on the type of cell, which makes sense because some cells are more active than others. So the number of chemical reactions could be anywhere between several hundred million to several billion chemical reactions per second. So let's say a reasonable average is about a billion chemical reactions per second per cell. Since by some accounts there are an estimated 37.2 trillion cells in the human body, then if we do the math, there could be around 37 billion chemical reactions per second. That's a lot of chemical reactions. And what's crazy is that each chemical reaction is catalyzed by an enzyme. Now, if enzymes did not exist, the chemical reactions in our body could still happen. They would just happen too slowly for our bodies to survive. So, because chemical reactions in our bodies must happen very quickly for cells to function properly, they need to be helped along, and that helper is an enzyme. Enzymes are what we call biological catalysts. What that means is that they help chemical reactions in living cells so that they happen much faster than they would without them. What enzymes do essentially is lower the activation energy at which a reaction occurs. This graph here shows how enzymes speed up a chemical reaction. Remember that in a chemical reaction, the reactants, shown here on the left, interact to form a product, shown here on the right. The wall that separates them represents the activation energy. You can think of the activation energy as an energy speed bump. The larger the speed bump for the chemical reaction, the slower the reaction. The yellow wall represents the activation energy of a chemical reaction without an enzyme, and the orange wall represents the same reaction with an enzyme. You can see the orange wall is a lot lower. This is because the enzyme physically brings the reactants together, and by doing so, it lowers the energy that is needed to get the reaction started, which essentially speeds up the chemical reaction. Notice that the enzyme does not influence the energy level of the reactants or the products, only the activation energy, the amount of energy that is needed to get the reaction started. So enzymes are proteins. Like all proteins, they have a very specific three-dimensional shape that determines their function. For enzymes, the 3D shape is specific to the chemical reaction it catalyzes. And enzymes have a place in their structure called the active site. This is the place where enzymes come in contact with the reactants from the chemical reaction. The reactants of an enzyme catalyzed reaction are called substrates. The shape of the active site is what makes enzymes specific to the chemical reactions they catalyze. And not all enzymes work on all substrates. The same way that locks and keys are specific to each other. Essentially, once the substrate or substrates attach to the active site, the enzyme provides the conditions for the chemical reaction to occur and form the product. And this is what lowers the activation energy. And it is very important that you understand that the enzyme itself is not a component of the chemical reaction. It is not a reactant, and it is not a product, and it is not used up in any way during the reaction. So an enzyme is the same molecule at the beginning of the reaction as it is at the end of the reaction, and can therefore be used over and over and over again to catalyze the same chemical reaction many times. How fast an enzyme works, its reaction rate, can be affected by the environment the enzyme is in. In particular is the effect of denaturation, a change in the shape of the enzyme. Factors which can influence the rate of an enzyme-catalyzed reaction include temperature and pH. So the rate of a reaction between an enzyme and its substrate is dependent on temperature. Let's start by looking at this graph that illustrates this. So in this graph, we have the temperature on the x-axis and the rate of chemical reaction on the y-axis. The rate refers to how quickly the enzyme is working. As the temperature increases, the rate of the reaction will also increase, but only up to a point, and then it's going to drop sharply right after that. So the optimal temperature is the temperature that occurs when the rate is at its highest. For a lot of human enzymes, the optimal temperature is around 37 degrees Celsius, which is body temperature. But the optimal temperature can vary depending on the type of organism. For example, there are species of heat-tolerant bacteria that live in geysers and other really hot environments, and their enzymes have an optimal temperature close to 80 degrees Celsius, which is almost boiling point. So obviously there can be a range in the optimal temperature, but most living things will find that their optimal temperature for enzyme reaction is between 37 and about 40 degrees Celsius. But let's explore why temperature has this effect on an enzyme's activity. What is happening in this graph? So first we saw that as the temperature increased, so did the rate of enzyme activity. And this is because as the temperature increases, so does the energy, more specifically the kinetic energy, of both the reactants and the enzymes. And this increased energy means that the substrates are more likely to encounter the enzyme, and to do so with more energy that allows the reaction to happen. So the higher the temperature, the higher the energy, and the higher the reaction rate. But when the optimal temperature is surpassed, the enzyme reaction rate drops sharply. And the reason for that is because at high temperatures, proteins can be denatured. And because enzymes are proteins, they also are denatured by high temperatures. When an enzyme is denatured, it loses its shape. And without the right shape, the enzyme cannot interact with the substrate anymore. So this is how temperature affects enzyme activity. At first, it speeds up the rate of enzyme reaction by increasing the energy that is available for both the substrate and the enzyme. But when it gets too hot, the rate of reaction drops to zero as the enzymes become denatured due to the amount of heat. Another factor that affects enzyme activity is the pH. So we have another graph, and this time we are placing the pH of the solution in the x-axis. Now enzymes also have an optimal pH upon which they work, which on this graph would be right here, where the rate is at its highest. And most enzymes work well at a pH value somewhere close to neutral, so close to 7, but somewhere between 6 and 8, most likely. But that's not the case for all enzymes. Some enzymes work well at much lower pH values or at higher pH values. For example, the digestive enzyme pepsin works best at the highly acidic pH found in the stomach, so it actually works best at an optimal pH of about 2. Another digestive enzyme works best at a slightly basic pH found in the small intestine. Now, both enzymes break down proteins, but work in different environments within the digestive tract, so have different optimal pH values as a result. So let's explore the reason for the shape of the curve for these graphs. And remember from our lesson on proteins that acids can also denature an enzyme. So as the pH drops, the enzymes stop working as well since enzymes are denatured at pH values lower than the optimal pH. And they are also denatured by bases at pH values higher than the optimal pH. So pH affects the rate of enzyme activity. And as you can see, there is a pH range upon which an enzyme works, but values outside of that range denature the enzyme, bringing the reaction rate to 0. And so that's it for the factors that affect the rate of enzyme activity. Something that you will get a chance to explore through an inquiry activity in the lab. And that's it also for our lesson on enzymes. Talk to you soon.